Okay, as we get started on this experiment, hopefully you've read the lab setup and you understand what we're trying to do for the brewery. We're doing an experiment that will help determine the best pH for making hard cider. As we get started on this, it's important to notice a few differences in our setup, our equipment that I've got set up here for you. You can see here I've got a few things that are different from what we originally started with, and we're going to go through what all these different items are. Here is our sucrose solution. It's already been made, 10% sucrose from the powdered form. It's just table sugar. And that would already be prepared for you. These are our fermentation tubes, and you can see that they're already labeled. A fermentation tube is kind of a strange piece of glassware. Notice it's open on this side and closed on the top. And we're going to fill that up with our solutions and trap gas in there. We've got our labeled beakers. These are small 100 milliliter beakers with stir rods, and they're all labeled. Over here we have some other glassware we'll be using, a couple of graduated cylinders, a large one and a small one, and a couple of beakers. Again, just general lab stuff. We also have our hydrometer. This is a larger hydrometer than what you've seen before, but it's the same thing. And again, it has measurements on the stem that will allow us to measure the density of a solution compared to water. Notice we're going to be using the graduated cylinder just to float the hydrometer. We're not going to actually measure volume with the hydrometer. We're using it for density. This is our universal indicator. We'll use that to measure pH. And here are our solutions for changing the pH. Citric acid, 2 molar, and calcium hydroxide. And they need to be shaken real well to make sure that they're going to be in good shape because they'll settle after a while. And then you see I've got whey boats with 1/2 of a gram of yeast in each whey boat. They have been measured out already. And yeast is a living organism. They come in packages like this. This is Fleischmann's active dry yeast. And you'll see that they're just little specks. They look like little dots. But those are clumps of cells that have been freeze-dried, but they are still alive and can be activated when given water and sugar. Part of your report is going to need to talk about what those organisms are and what they're doing. So those are yeast, 1/2 a gram in each whey boat. Please make note of the changes that you see here. These are really important to write down because when you write this up in your lab paper, you will need to follow the procedure that you see listed here and that I will be demonstrating. Please make sure that you're following this and not what's in the lab manual for the first 9 steps. Okay, we're ready. I've already done the first few steps, so we're ready to get our 200 milliliters of sucrose solution. So I've got my beaker. All we need is 200 mils, so we're going to use a large beaker. And there's our solution. The glassware is already labeled and the yeast is already weighed out, so we don't have to worry about those first steps right now, but make sure you note them in your manual. All right, there's 200 milliliters of sucrose. Now we need to add 5 milliliters of our universal indicator. So I'm just getting that. The universal indicator changes color with changes in pH. So kind of like some of the other indicators we've used, it's an easy way to determine pH across a large range. There's our 5 milliliters, and we'll dump that in, and you'll see that that turns our sugar solution, or our sucrose solution, green. Okay, and I'll hold that up so you can see it as it's kind of swirling around in there. Notice with a white background you can tell it's pretty green. And now what we need to do is put that in our beakers and modify the pH before we put any yeast in there. As soon as we put the yeast in with the sugar, it's going to start fermenting and it's going to mess up our results. So we don't want to do that too soon. We need to go ahead and get our beakers set up with the right pH first, and then we'll add our yeast last. So I'm going to get my small beakers ready and put them in order. And we're going to create a gradient of pH from beaker number 1 to beaker number 5. So we're going to add 40 milliliters to each beaker. And again, it doesn't have to be perfect here, it just needs to be pretty close. There's 40 milliliters. We'll do that for each one. And then once we get these set up, notice that it is beaker number 3 that we're not going to change. That would mean it's the control. Okay, so there's our beaker number 3. Number 1 and 2 will be more acidic. Number 4 and 5 will be more basic. And 3 will remain close to neutral. Okay, so we've got these ready. And now, as you can see, they're green, so this is a neutral pH. And if you can see the colors and numbers, it's a little blurry, but you can see that right now they're at about a pH of 7. And what we want to do is make some of them acidic and some of them basic in sort of a range. And we're going to modify these using citric acid for the acids. That's going to be beakers 1 and 2. It's a 2 molar solution. And we want beaker number 2 to be somewhere around a pH of maybe 5. It doesn't really matter as long as we create a gradient, but that's what we're shooting for, just as a lower pH. And you can see there the color changed very quickly with just 1 drop. That probably brought our pH down to 6. I think I want to go a little bit further though. Let's see if we can get a little bit more of an orange color. Right now you can see it's pretty yellow, maybe a little bit more orange than yellow, but starting to turn orange, but not quite there yet. So I'm going to add another drop, see what that looks like. It doesn't take much. Okay, and that's a nice orange color there. So if we hold that up next to the card, you can see that's about a pH of 5. I'd say that's pretty close. Okay, and we may want to go ahead and jot that down so that we're sure we know what we're doing here. That's a pH of 5. Alright, what we want to do on this one for beaker number 1 is get it even more acidic and get our pH even lower. And we're going to shoot for 4. So we know we're going to need a couple of drops. And there's 1, 2. Let's see what that looks like. Nope, that's already a little bit more red. Those are a little bigger drops. Let's go ahead and add a little more and see if we can get it a nice bright red. Oh, there we go. That looks good. So that's definitely a nice red color. And if I hold that up, you can see that that's about a pH of 4. With the white background, you can tell pretty easily. That's about a pH of 4. And we'll go ahead and note that as well. And remember that beaker number 3 started off at the beginning as just that green color, which we know is a pH of 7. That's our neutral pH. Now we're ready to do 4 and 5. We're going to do them the same way, except now we're making our pH higher by adding a base. And this is a 0.3 molar solution of calcium hydroxide. Again, just a drop at a time. And it's kind of hard to see the color change when you're going from a green to blue or kind of a teal color, but I don't know if you can see that. That's probably a pH of 8. That's a little bit more of a blue-green color. I would say that's close enough to 8 that we can go ahead and call that 8. And for our last one, let's just take that all the way to 10. So for that one, we'll need at least a couple drops. And a pH of 10 will make it purple, so we're shooting for that nice purple color. And a couple more. And there we go. It's getting definitely a kind of a brighter bluish purple. Add a few more drops there. Okay, and that has definitely turned purple. Okay, and if I hold that up next to the card, you can see that that's definitely a pH of 10. Okay, so with that pH now at 10, we have a continuum of pH ranges from 4 up to 10, with our control being 7, which is neutral. Okay, now what we need to do is we're going to add the yeast to our 5 beakers, but we need to do this quickly because as soon as we put the yeast into the sugar solution, we have to put it right into our fermentation tube because fermentation will begin. And we want to put the solution in, we're going to tip it, and when we tip it, it's going to fill this vertical part of the tube, and then it will catch the carbon dioxide produced by the yeast in the vertical part of the tube. The volume of carbon dioxide will indicate how much fermentation is occurring. So I'm going to dump these in and stir them up real quick. And this is one of those things we want to do, try to do it rapidly so that we don't let those yeast start to ferment too quickly. If you remember, carbon dioxide mixed with water, which obviously this is a water solution, does produce carbonic acid. So you're going to see as yeast begin to break down the sugar and produce carbon dioxide, the solutions are going to change color as they become more acidic. So don't be surprised when you see that they all start to shift a little bit in a more yellow or orange or red direction as the yeast get busy. But it was the starting pH that mattered. That's our original question that we're trying to answer, is what starting pH will give us the best results. Okay, so now that we've got those in there, we're ready to fill up our fermentation tubes. And we'll just do this again quickly so that we can then get the fermentation tubes into the incubator at about the same time. So notice that I'm just going to pour the solution in. Your procedure says that you have to measure it out, but it really doesn't matter because the amount that we have in the beaker is just about the right amount. Now there's some yeast left behind, so I'm going to swirl that around to make sure we try to get all the yeast out of the beaker. We want to make sure that it's pretty empty and our solution is going to have all the yeast in it. So when I tip it, notice that it fills up that vertical part of the tube. When I bring it back up, it stays in that vertical part, and now it will start to capture any bubbles produced. Okay, we'll do that with number 2. I'm going to go through these real quick, and you can see they're already starting to change color a little bit. The yeast are already becoming active. Doesn't take long. Okay, and now we're ready to stick these guys in the incubator. Need to do that real quick, so I'm going to go ahead and set them in there and be right back. Okay, so I forgot to do something before we stuck our solutions in the incubator. We need to measure the specific gravity of our initial solutions. So I've got a remade sample here. This is just our sucrose solution, yeast, and indicator. I did not change the pH, but we just need to measure the specific gravity of our starting solution. It'll be the same for all of our 5 initial fermentation tubes. And it looks like our initial specific gravity is 1.030. It's too hard to really show you that, but if you see the numbers on the stem there, they're very, very tiny. But if you look at where the level of the liquid and the stem meet, it's 1.030. And that's going to be the same for all of our starting fermentation tubes. Okay, this is our incubator, and you can see that the fermentation tubes are now in there. They've only been in there for a couple of minutes, so there's really no gas collecting yet, but we're going to wait and give them about 45 minutes. Notice the incubator is set at 45 degrees Celsius. Since it was just opened, you'll see that the number says 42, but it's set at 45, and we'll let them incubate at 45 for about 45 minutes. Alright, so now I have gathered them from the incubator, and let's take a look at our results. And we need to do this quickly again because the yeast are still active and they're still making carbon dioxide, but we want to mark them all at the same time. So notice I'm just using a wax pencil and I'm marking the level of the liquid. The stuff in the top of the fermentation tube is just some foam that was produced, so that's really not important for us. What we want to see is where does the level of the liquid now sit, and the carbon dioxide that was produced by the yeast has pushed that liquid down as the carbon dioxide has collected in the top of the tube. Okay, and you can see it's fairly easy to just mark right where the level of the liquid is with the wax pencil. Now it's important to understand a few concepts as I'm marking these. The more the yeast have done some kind of cellular respiration, either aerobic or anaerobic, they are going to produce carbon dioxide. So the amount of some kind of metabolism is measured by the volume of carbon dioxide. But the question is, are they actually doing fermentation, which produces alcohol? The more sugar that's turned to alcohol, the lower the density, because you may know this, alcohol has a lower specific gravity than water. Alcohol is lighter, it will float on water. So we're going to use the smaller hydrometer just to deal with the volume. I'm switching from our large hydrometer to the small one to be able to measure the change in density, which will tell us the alcohol content. Now, these have only been going for 45 minutes, so we're not making booze here. This is not going to be a lot of alcohol. It's a very, very small amount. But we're actually measuring our fermentation by two different variables. One will be our specific gravity change. The more the specific gravity changes, the more alcohol was produced, but also by the volume of carbon dioxide produced. And that may have been produced aerobically or anaerobically. It's important to understand this concept when you write this up in your paper. All right, it looks like that first specific gravity for our first tube is 1.026. 1.026. So make sure you write this down in the data table that accompanies this. Okay, and I'm just going to dump out the liquid because we're now done. We don't need to worry about saving the liquid anymore. And I'm going to rinse off my hydrometer with some clean water. And then we'll do the same thing with our other 4 fermentation tubes. You'll notice that as I'm doing this, the gas continues to be produced and the liquid keeps going lower. But since we've already measured the volume of gas, we don't need to worry about that. Okay, so I'm just going through the rest of our tubes. You see the data listed there. Tube 2, 1.024. Tube 3, 1.028. Tube 4 is 1.026. Tube 5 is 1.026 as well. So understand that what we're really looking for in a change in density is how much sugar has been used and how much alcohol has been produced, and that's what's changing their density. Okay, now we're ready to measure the volume of carbon dioxide produced by each of our fermentation tubes. So I've dumped all the solutions out, rinsed out most of the foam. What we want to do is figure out how much gas occupied the top part of our fermentation tube. Now, that's really hard to measure because you can't pour out gas into a beaker or a graduated cylinder, but water can. So we just want to add enough water to match that line and then pour that water into a graduated cylinder, and that'll tell us how many milliliters of carbon dioxide were produced. So notice all I'm doing is putting enough water in there so that when I turn it upside down, the amount of water will match the line that we drew. Okay, and I'm kind of playing around with that because it's tricky. We want to try to get it as close to the mark that we've made as possible. Okay, and that's pretty close. It's a little hard to see at the angle the camera's in, but the meniscus is actually at the red line if you had it at eye level. It looks like it's a little bit above it when you're looking down on it. But now what we want to do is just find out how much that is by pouring that liquid into a graduated cylinder and then measuring the volume on the graduated cylinder. Okay, and for our first fermentation tube, our volume of carbon dioxide produced was 11 milliliters. Okay, our next one, we'll do the same thing, and I'm just going to speed this up a little bit. Next one is 14 milliliters. 14 milliliters. Tube number 3 will be 9 milliliters. Again, we're just replacing what was gas with water and then measuring the volume of water. Okay, so 9 milliliters. Tube number 4 is 10 milliliters. And tube number 5 is 8 milliliters. Again, these are two different ways of measuring what's happening in the tubes. One of them is the amount of carbon dioxide produced. The other is the change in specific gravity based on the use of sugar and the production of alcohol. So your paper needs to talk about both of those sets of data. Okay, so now that we've got our data, what you need to do at this point is think about what are yeast, what were they doing, what variable did we manipulate, what variables did we measure, and what do they mean? So now it's up to you to analyze and explain the data and come up with some conclusions and write it up as though you were writing a serious scientific paper. And we got to use some cool equipment called fermentation tubes to do this. So hopefully you had fun and you learned some things and can write it up.